In this video, we're going to be discussing IMViC testing with E. coli. For this experiment, we're going to be on day 2, which is after 24 hours of inoculation. Add 0.2 ml of COVAX reagent to your tryptone broth tube. Gently mix your sample. Here's a close-up of the completed indole test. For this experiment, we'll be on day 3, or 48 hours of inoculation. Remove 1 ml of culture from your MRVP tube and place it in a sterile tube. Get an ampule of alpha-naphthol or Vogue's Proskauer reagent A. Break the glass rod in the ampule to release the liquid. Add the contents to the 1 ml broth. Do the same for the 40% KOH or Vogue's Proskauer reagent B. Gently shake the tube to mix. A fresh MRVP tube is on the left and our sample is on the right. Remember that a positive result should be bright red. The following experiments will be conducted on day 5, or after 96 hours from inoculation. Add 250 microliters of methyl red to the MRVP broth culture. Here is a close-up of the result from the methyl red test. Remember that the bright red is a strong positive result. A bright orange color is considered a weak positive, and a yellow is a negative result. We also need to check our Kosar citrate tubes for turbidity. Our inoculated tube is on the left, and a fresh tube is on the right. We also need to check our BGLB and E. coli broth tubes. Remember that by day 5, they've been inoculated for 48 hours. Positive BGLB tubes will have gas production in the Durham tubes. The far right tube is a fresh tube, the rest are inoculated. Positive E. coli broth tubes will have gas production in the Durham tubes. The far right tube is a fresh tube, and the rest are inoculated. Positive E. coli broth tubes will also fluoresce under a UV light. You can see the full results from this lab in your Blackboard folder. You should use these results to calculate your results for your lab notebook.